Hi class, this is your instructor, Skylar Huff, and welcome back. We're now in Chapter 6 with cell communication, and this is the final chapter of your very first test. To maintain homeostasis, the cells of multicellular organisms must continuously communicate with one another. Over billions of years, there have been many different mechanisms for transmitting information between cells, tissues, and organs to have evolved. And this does include that electrical signaling and even many types of chemical signaling. So in addition to that, there are organisms of different species and even different kingdoms and domains communicate with each other. So the organisms, they can communicate with other members of other species by secreting chemical signals. For example, bacteria release chemical signals that diffuse among and attract nearby cells. As the population of bacteria increases, the concentration of the chemical signal also increases. It's through the process known as quorum sensing. The bacteria will sense when a certain critical concentration of a signal molecule is reached and respond by activating a specific biological process. For example, they may form a biofilm, which is a community of microorganisms attached to a solid surface that requires the coordinated activity of numerous bacteria. So there are many chemical reactions that are involved in responding to signal molecules and regulating communication among cells. So many researchers work on the molecular level to understand how individual proteins receive messages and relay signals. They are learning how molecules act as switches, activating and deactivating complex signaling pathways. Other scientists, however, use a systems biology approach to understand the intricate dynamic interactions involved in cell communication using methods derived from biochemistry, biophysics, and genetics, in addition to other disciplines. So when they, of course, work to understand how elaborate signaling systems within the cell interact to regulate its functions. Through these studies of how information is transferred between cells in tissues, organs, and throughout the organisms, scientists have shown that faulty signaling mechanisms can cause a variety of diseases. For example, many forms of cancer, now known to result from genetic defects that alter signaling systems responsible for cellular growth and division. The increased understanding of these cellular communication mechanisms has suggested new strategies for preventing and treating these diseases. So as we continue on, what I have before you, class, is figure 6.1 from page 132 of your textbook. If you look closely, what we're seeing here, class, is cells signaling in cellular slime molds. We have not yet learned about slime molds. However, in our last chapter for study, when we get to Kingdom Protista, we'll get to slime molds. So when a food source is scarce, these amoeba-like slime molds, they secrete that chemical compound Cyclic AMP or CAMP. The slime molds respond to this chemical signal by aggregating. So what they then do is, of course, converge in streams of hundreds of individuals that come together to form a multicellular colony. So as we continue throughout the chapter, please make sure you take really good notes, and if you have any questions, feel free to email me. So let's begin now, class, with the way in which species come together. And I mention this because species must communicate. And even, as I mentioned a bit earlier, class, that organisms of different species can very well communicate, such as the example being shown here with the seaweed class and bacteria. So it's not just our cells communicating with each other within our body. It also includes, class, organisms of different species. So as you are, class, going through what you should know for your very first test from Chapter 6, please restrict that class to section 6.1 from pages 132 to page 133. I'll now continue, class. So when I get to this, class, what you are responsible for is right here with this overview class of cell communication. So I'll begin class with what is known as cell signaling, and it refers to the mechanisms by which cells communicate from, of course, with one to another. So if the cells are physically close to one another, a signaling molecule or a ligand or ligand class, and that's spelled Ligand, a ligand or a signal molecule, the ligand. So what happens, class, is they may combine class with a receptor. And when you think of a receptor, I want to write the term receptor here. Think of it, class, being possibly a protein. It may very well be, class, a receptor protein class embedded within the cell membrane. A protein class embedded within the cell membrane. I guess I'll put the word C-E-L-L, cell membrane. And of course, it may bind, or rather combine, class with that receptor, which it states, class, a macromolecule, which proteins are macromolecules made up, class, of those small repeating units known as amino acids. So most commonly, class, cells communicate by sending chemical signals, class, over some distance. So as we continue on here, class, throughout the chapter, just keep in mind that cell signaling must be precisely regulated. It's not just something that happens, class, by chance. It's precise regulation of such, and I'll continue to give you all examples of this throughout this lecture. So with this, cell signaling involves, class, a four main sequence, or at least a four process sequence. And those four processes, class, are shown here before you. So first things first, class, it involves signal transmission, reception, class, of that signal, transduction of the signal, class, and then ultimately, class, some cellular or physiological response. So let's begin now, class, with signal transmission, the first of four main processes. With signal transmission, it's happening, class, in chemical signaling. And what I mean by this is a cell must be able to synthesize. It must be able to make and release those signal molecules or those ligand molecules. So I'll put, class, again, Ligand. The cell must be able to synthesize those. So as it happens, it could very well be, for example, class, happening in specialized cells of the vertebrate pancreas. I did say, class, in the specialized cells of the vertebrate pancreas. So as I say it that way, we all have a pancreas, class. So in the pancreas of which, class, it may very well be that the hormone, class, insulin is secreted. So I'm going to write here, class, to make it a bit more specific, it would be those beta. It would be beta cells, class. Beta cells of the pancreas are what, class? Secrete the product. Secrete, class, the hormone, insulin. And I'm writing all over everything right now, so I hope you can follow me. And if you can't, just let me know. So from that, it states that if the target cells, and keep in mind, class, the signal being secreted, being released, class, is insulin, that endocrine hormone. So if the target cells that respond to that signal, or any signal at all, are not in close proximity, class, that signal must then be transported to them. So by way of our circulatory system, class, it transports insulin to the target cells throughout the body. And this is where things, class, are so amazing. So from that happening, class, it's next that those target cells must then receive, 1, 2, relay, and 3, respond, class, to that information that was ultimately signaled to start this entire cascade of effects. Next up, class, is reception. We're now here. So with reception, it's the process of receiving that incoming signal, or receiving, class, what the ligand or a ligand, class, is attempting to convey to the rest of the body or the cells by way of course, that message. So the receptors are large proteins. I mentioned that just moments ago. If they're not proteins, they're glycoproteins. And they are what bind to that ligand, or what bind, class, to that signal molecule. So many types of signaling molecules do not, I repeat, many types of signaling molecules do not enter the target cell. They bind to specific receptors on the surface of the target cell. So let's just say this is some quote-unquote cell. Hi, cell. And let's just call this a eukaryotic cell. So what I'm getting at is on the outside of that cell, there may be some epitope or receptive region. So as it says right here in your text class on page 132, right-hand column class, number 2. As they bind, and in this example class, insulin is used. What then happens, class, is that it binds to those insulin receptors, which are transmembrane proteins. So I'm going to make this make a bit more sense. Think of it as being this way. You have a body, I have a body. Hey, we both have bodies. So in the way in which it works is that we eat. Oh yeah, I said that. So upon eating, whatever you eat, class, has those building blocks for life. And I don't mean Legos. I'm going back to a prior chapter class, referring to, of course, your carbohydrates, your lipids, and your proteins. In this case, we want to restrict ourselves, class, to insulin. So what happens is, at some point, class, you need energy. By the process, class, of respiration, referring to cellular respiration, what happens, class, is you take what is called glucose, and you use that glucose, class, to synthesize energy in the form of ATP. So as you eat, as you chew, by way of, it's called mastication, you break down, class, those larger, I guess I'll call it food particles, those foodstuffs, into smaller foodstuffs. Well, what happened was, give me a little space here. Let's say this is me and this is you. All right. I guess that would be an ear here and eyes there. And maybe you have on a cap because it's summertime. Alrighty, so there's a neck and boom. I don't know why I keep shooting off those lines there, but thanks a lot, computer. Alright, so cross section here. So I'll restrict myself to this. We eat. As you eat, and of course you swallow, here's that stomach of which, here's the small intestines. I don't know why those lines keep getting thrown out. Here is the large, and boom. I guess we don't have a brown color here, but let's make it happen. So what I'm doing here, class, is getting back to my example. You've eaten. You've chewed the food up, class, going through the process of mechanical digestion. And now let's begin the process, class, of chemical digestion. So by way of chemical digestion, class, the foodstuffs ultimately make it into your stomach and even, class, into your small intestines. Well, by way of your small intestines, class, you absorb those macromolecules. You absorb, class, those I guess you say smaller substances, class, that make up the macromolecules, such as, class, what are known as monosaccharides, such as, class, I guess you say the glycerol or the triglyceride or those fatty acids or even, class, those amino acids. As you take those things in, class, you cannot do a thing with them unless first things at first, class, you have what is known as, grab the other pen here that is that color. Oh, the pen is ready to go. Unless, class, you have insulin. So keep in mind, I don't know why that L looks like that, but that's insulin. Unless, class, you have insulin from, of course, what is known as the pancreas. Unless you have, class, insulin from the pancreas by way of the beta cells, you cannot, class, use the glucose. It is insulin, class, that allows glucose entry into, class, our cells. Without insulin, class, glucose cannot enter into those blood cells. And of course, your blood glucose, class, would remain, of course, where it is, unless insulin, class, is allowed entry by way of course, unless glucose is allowed entry by way of what you see right there, class, known as insulin. So it's insulin, class, that is known as that key that unlocks, class, what I call sugar's access, or the entry, class, of sugar into the cell. So what I'm getting to is it's the, of course, signal molecule that has to be there first. And then reception class of that signal that then class allows glucose entry. I'll continue now class with number 3. So next up class is signal transduction. With signal transduction class, this is the way in which your cell converts that extracellular signal class into that intracellular signal or set of signals. And this causes class that response. I once used class kind of like your phone ringing, if ever your phone rings. I'm hoping your phone does ring at least once, sometime. So when your phone rings, class, you can think of that as being, of course, that signal. Then, of course, you pick up the phone because the phone's vibrating. And that's reception of the signal. Then someone says something, class. And, of course, as he or she says what they say, class, that would be transduction. Meaning, it typically, class, involves that chain of molecules that relay information, such as those words, class, being said to you. And then when insulin, class, as I just mentioned, bind to the insulin receptor, class, on, of course, the outer surface of the cell, the signal is relayed, class, through several different signaling mechanisms. Now onto the response, class. So with the response, or by way of the response, the final molecule in the signaling pathway is what converts our signal into a response that alters some cellular process, some physiological process. So, of course, insulin, class, it says stimulate cells to uptake class glucose from the blood. I repeat, it stimulates our cells to take up glucose from the blood. So I guess I'll put it as A, glucose. Whereas glucose, class, is in those biconcave red blood cells. It cannot, class, be used unless first things first, insulin, class, is received. That's insulin is received to allow, class, that glucose to be taken up by our other cells for use to, I say, ultimately power everything you do. From you hearing my voice now, to you sitting in that chair now, or sitting on that bed now, or even, class, sitting in a lecture right now. So in what I'm giving you here, class, it just lowers the concentration of glucose in the blood. That way, class, you're not hyper. That way, you're not, class, hyperglycemic. Hyperglycemia, class, can kill. And in some cell types, class, insulin is also involved in the regulation of fat and protein metabolism, meaning I just alluded to that, class, moments ago. So with this, it's many signaling molecules that stimulate ion channels in the cell membrane to open or close, and still other signaling molecules activate or inhibit specific genes in the nucleus. So these responses can result in changes in cell division and other aspects of development. So after a signaling molecule has done its job, class, its action must be stopped, and it is part, class, of that signaling process that certain mechanisms operate to terminate the signal. So as far as your test is concerned, you can also look at figure 6.2 on page 133, because this figure, class, it provides an overview of what I just did. For what reason, I don't know, but that presentation, class, was not included in the PowerPoint as you see here, but I did do a bit of illustration, class, moments ago. So on your test, class, these are things to know. Now let's continue on with a bit more biology. I repeat, on your test, I'm testing you all, class, on section 6.1. This further information, class, is things you all ought to know. Let us begin. So here, class, we get to, of course, the sending of signals, meaning this is on page 133, right-hand column, class. Let us begin. So cells that are not directly connected, class, also communicate with one another. In animals, class, some neurons communicate with electrical signals. And what I'll say is that most neurons, however, as you all see down at the bottom of the screen, most neurons signal one another by releasing chemical compounds called neurotransmitters. Yes, neurotransmitters. So briefly here, I'll draw neurons. So that's a dendrite, that's a dendrite, dendrite, dendrite. Now I'll draw, class, this long axon. This is the axon with the synaptic knob. That, class, is known as the nucleus, which is their, class, within the cell body. The space, class, that I'm leaving between the two is known as the synapse or even the synaptic cleft. I'm going to draw a bit more here. I'll stop for a moment to think. I'm done thinking. So what I'll now draw here, class, is going to be 3 skeletal muscle fibers. I'll now add the striations class to those skeletal muscle fibers because skeletal muscle class is striated. I wanted to erase that. It did not look good at all, so I erased it. Maybe I should slow down to make sure it doesn't give me those lines off to the side as it did earlier. So we have 1, 2, 3 skeletal muscle fibers here that are striated because skeletal muscle class is just that. It is striated. And now I'll add class here in the periphery, the nuclei, meaning skeletal muscle fibers class are multinucleate. That's why you're seeing class many different nuclei per muscle fiber. And I'll notice I'm not calling these skeletal muscle cells, I'm calling these skeletal muscle fibers. So having drawn that, what I'll get to now is, I guess I'll say, meat and potatoes. Let us begin. So we have the neuron here, class. And I'll be blunt, the synapse or synaptic cleft class is that space between the neuron class and this skeletal muscle. In your text class, it says neurotransmitter molecules diffuse across synapses, tiny gaps between neurons, or at least between the neuron class and, of course, that junction with that skeletal muscle. So what happens is this neuron class receives some stimulus. That's reception. So as that stimulus class has been received, I'll make sure that you all know that the action potential will go class in one direction. The action potential class will go in one direction. So as it goes class from, of course, the dendrites, which will receive that stimulus, down class the axon helic, and then down this axon itself, as that electrical message class makes it to that synaptic knob, what then happens is that there's a change class in voltage, kind of like we've done before. So time is class here at the X, time is here class at the X in milliseconds, and then voltage class is here on the Y. We're at a negative 70 here, we're at that positive 30 there. I wrote 30 there, literally. Let's begin. So what I'm saying there at the top right class in green is that action potential here at this overshoot is that reversal from that slightly more negative to that positive class charge here inside the neuron. The neuron class is to the left in purple. So what happens is that stimulates class voltage-gated calcium channels to open. And I'll get to that a bit more. So when those voltage-gated calcium channels open, class, it causes vesicles of acetylcholine, the neurotransmitter class, to form, which I'm drawing here, class, in a kind of green. I got 3 green lines or dots. So what then happens is acetylcholine will diffuse, class, across the synapse and bind to its receptors here on this skeletal muscle fiber. And what will then happen, class, is the very same will occur in the muscle, that it will go through the process, class, of eliciting, class, an action potential, and the ultimately, class, causing contraction. And this all because, of course, this neurotransmitter class called acetylcholine. I'm writing ACH, but write it out, class, as you see it in your text, acetylcholine. There it is. That, class, is a type of neurotransmitter. It was the first discovered, and I say the most well understood. Now back to reality. So as I'm back here, what I'm saying about this, class, is this is just one of 60 different types of neurotransmitters that have been identified. And that includes, class, both epinephrine or norepinephrine, dopamine, serotonin, a number of amino acids, and peptides. So cells can synthesize many different types of chemical signals and deliver them, class, in a myriad of ways. So in certain animal cells, the immune system produces specific chemical compounds that are displayed, class, on the cell surface. For instance, I'll mention the term here, class, that you may or may not remember. I'm saying epitope, meaning cells may have an epitope on the outside, that receptive region that's very specific class and specific to a specific type of course, signal molecule. So as they are displayed, the cells recognize the chemical signals class and then of course communicate with each other and making class direct contact. And so as you're seeing here on the screen, class is kind of a lock and key fit. Next up, it states that of course cells class can also communicate in, I say, a different way. Let's say this way, class, by some chemical signal, meaning hormones. So hormones may be synthesized by neighboring cells or specialized organs or tissues. It states here, class, some distance from the target cells. So in animals, many hormones are produced, class, by endocrine glands. The endocrine system, class, is I say, one of the smaller systems you'll ever learn about. No, I'll take it back. It is the smallest system you'll learn about. You can fit all those endocrine class glands in about the palm of your hand. But of course, class, if one of which was missing, class, it would be strikingly obvious, class. I would say not just, of course, I say physically, but class, most definitely physiologically. So these glands, the endocrine glands, have no ducts. They're ductless glands, and they secrete their product, class. They secrete those hormones into the surrounding, class, interstitial fluid. And it's typical of these hormones to diffuse, class, into capillaries, or even, class, I guess you say, and are transported by blood to the target cells. So you're seeing, class, those hormones here in green. I guess I'll type out or write out, class, the word hormone. And hormones, class, are here in green. That, of course, bind to their very specific receptors, class, on those target cells. So some cells produce, well, I'll get to it, local regulators. And in getting to those local regulators, let's get back to one last thing. Meaning it states that those local regulators that diffuse through the interstitial fluid surrounding the cells, they act on nearby cells. So the process is called paracrine regulation. So those local regulators are considered hormones too. And they include things, class, known as those local chemical mediators, such as growth factors, nitric oxide, prostaglandins, and even, class, histamine. And let's start here, class, with histamine and its effects. So histamine is a local regulator that is stored, class, in certain cells of the immune system. And as I say it that way, you can also think about, I guess you say, mast cells, which you haven't quite learned about yet, but mast cells can also be here responsible, class, for the release of histamine. So it is released, being histamine, class, in a response to an allergic reaction, which I hope you all have not had yet, or if you had it, I hope you had an EpiPen. Thanks a lot, epinephrine. Or I just hope you never have one if you've not had one before. Well, like one time, I wasn't at band camp, but one time I had, I guess you would call it, an allergic reaction. I was cleaning shrimp. And I'd always clean shrimp as a young boy. My dad would say, go in there, and my brother and me, we would clean shrimp, standing in the kitchen in Wiggins, Mississippi, at home. Well, this time I was an adult at my house, cleaning shrimp, pounds and pounds of shrimp. But then I began to have an itch. I was definitely, class, itching for a scratch. It was behind my knees. And I was using my foot because I was cleaning shrimp. You know, dirty fingers, you're not going to reach back there with a shrimp finger. I kept cleaning shrimp anyway. But then it got to the point where I just couldn't bear it anymore. So I walked to the bathroom and washed my hands, and I began to feel that it was hard to breathe. I then called said spouse, and I said, hello, it's hard for me to breathe. And I was then noticing, hello, you're having an allergic reaction to the shrimp you're cleaning. And I was cleaning shrimp for a very long time. I said pounds and pounds of shrimp. So it was because of, class, that release of histamine. So I duly, class, took what is called, if you haven't heard yet, an antihistamine. You may sometimes call that antihistamine, class, that you all know. Come on, help me out. The antihistamine that may be in your cabinet. I'm waiting. Okay, I took a Benadryl. That's the name of the antihistamine. Meaning I'm sure you all knew about it. But yes, I took a Benadryl, class, and went right to the emergency room. And I put an IV was put in my vein, class, directly to ensure, class, I did not die. So please do not take class allergic reactions lightly. Back to my story. So histamine class could be released because of some injury to you or even class an infection. So getting to how histamine class affected me or affected you or will affect you or I guess you say can affect you is first things first. It causes class blood vessels to dilate. Meaning, let's say that is what is called a blood vessel. So what I'm referring to, class, is the lumen. That's Lumen. The lumen, class. That looks ugly. But I'm referring to, class, the lumen of the blood vessel. So if you look there, that is the lumen of the blood vessel, class, and I'll say here when it's normal or at homeostasis. I like that word, homeostasis. Well, I said that histamine causes blood vessels to dilate. So this, class, is now that dilated blood vessel. And it's all because of the release of histamine, meaning the response to it. So it dilates class blood vessels and causes capillaries to become more permeable. Whereas nitric oxide, class, another local regulator, part of course, paracrine regulation, is a gas produced by many types of cells, including class plant cells. So nitric oxide is released by cells lining blood vessels, and it too, class, relaxes smooth muscle in the blood vessel walls, resulting in which, class, blood vessels then also dilate, class, causing a decrease, class, in blood pressure. That's BP. So with prostaglandins, I'll get to now, they are local hormones that act as paracrine regulators. So prostaglandins can modify cyclic AMP, CAMP levels, and then of course interact class with other silly molecules. For example, it may be prostaglandins, that some of which, class, stimulate smooth muscle contraction. So let's get now, class, to reception to make this make a bit more sense. So with reception, there are hundreds of different types of signaling molecules that are present, class, within our interstitial fluid and the tissue fluid that surrounds our cells of course, the multicellular organism. So the way in which our cells know which messages are for them, class, is that each type of cell is genetically programmed to receive and respond to specific types of signals. So it's not one signal for all. They're very specific signals. So which signals a cell responds to depends upon, class, those specific receptors. I repeat, those specific receptors it's programmed to synthesize, programmed to make. And as stated before, class, it's these ligands, it's these signal molecules, such as insulin, that bind to those specific receptors that act as that, of course, signal. So a ligand, as I've been saying, class, the entire lecture, L-I-G-A-N-D. A ligand is that molecule, other than, class, an enzyme that binds specifically to a macromolecule. And, class, it's typically a protein. That protein that's, of course, within, class, the cell membrane. And as I say it this way, it's forming that macromolecule ligand complex, or that signal or ligand protein complex. They're as one. I'll put it that way. So most ligands, class, as it states, are hydrophilic molecules, most of which are hydrophilic molecules, class. And with that they bind to the protein receptors on the surface, class, of the target cells. And in the event, class, that it was hydrophobic, meaning it's because of hydrophobic molecules, those, of course, would move, class, directly through the plasma membrane and be able to bind with, class, some intracellular receptor. So that comes back from what you all learned about plasma membranes in the prior chapters. Let's continue on. So as it states, class, if in fact that silly molecule fits that specific shape of that specific receptor, class, it can most definitely trigger, class, that cellular response in the cell. I told you, class, it's very specific. So different cell types produce, of course, as I mentioned, different receptors. And I say that's enough about this. Let's get to regulation, and we'll be done with regulation, class, here shortly. This is on page, class, 135. So here, class, it mentions that an important mechanism that cells use to regulate reception is increasing and or decreasing the number of each type of receptor. So let's go to upregulation first. So with upregulation class, it occurs in response to a low hormone concentration. So in this way, a greater number of receptors class are synthesized, and their increased numbers class on the plasma membrane make it more likely that that signal will be received by a receptor on the cell. Thus, upregulation class amplifies. Thus, upregulation amplifies class that signaling molecules effects. To contrast, let's get now to downregulation. So with downregulation class, what happens here is the concentration class of, or at least the number of those receptors class are destroyed by way of course, those lysosomes. So those lysosomes class are going to be transported to those receptors class are going to be transported to the lysosomes, where they are then class destroyed. And to make it make a bit more sense, I'll put it this way. In our example class using insulin, it says here that the receptor downgrade regulation process suppresses the sensitivity class of the target cell to that hormone. So insulin class would stimulate the cells to take in glucose by facilitated diffusion. I cannot stress enough, it is insulin class. Please get this. Thank you. It is insulin class that stimulates our cells to uptake what is known as glucose by way of facilitated diffusion. And what I'm saying here, class, is by downregulation, it would involve class, of course, the exact opposite, so that the uptake of glucose class happens to be slowed or ceases class completely. So now we're getting to ion channel link receptors. So these ion channel link receptors class, keep in mind that these receptors have been extensively studied in neurons and muscle cells. And I'm going to go back to what I did earlier class, and I just might not take quite as much time here. So I'm going to pick up my pace just a bit, because I think you all are pretty much with me now. All right, so here is some motor neuron. Here is the motor neurons class nucleus that's there in the cell body. Of next class, I'm going to draw, yet again, class muscle fibers. And I don't know why that just did that. I guess maybe my finger didn't pick up off the screen quick enough. Back to work. So here we go. So I have 3 muscle fibers there. Three muscle fibers are here. I'll now class, after now drawing my striations, I'm going to draw my purple staining nuclei that are there on the periphery of the cell. I'm going to draw my purple staining nuclei that are there, class, on the periphery of the muscle fiber. I said cell, muscle fiber. So I have 3 muscle fibers here, and I'm drawing my nuclei that, of course, accompany those muscle fibers. All right. So to get to ion-linked receptors, or ion channel link receptors, what I'm getting at, class, is those ion channel link receptors are also called ligand-gated channels, meaning that the ion channel class opens and closes in response to binding of the signal molecule, the ligand. So, nerve impulse class, let's begin. We go in one direction, class. So there is a receptor, meaning the receptive region class of the neuron being the dendrites. The dendrites class are the receptive region. Of course, the nerve impulse class goes down this long axon that is there. And then ultimately, class, what happens is, in this case, we get to class voltage-gated calcium ion channels. Voltage-gated calcium ion channels. They open class in response to the change in voltage. Calcium class enters into the synaptic knob. Thereafter class, immediately thereafter, we get, of course, the neurotransmitter acetylcholine. It then class will diffuse across the synaptic cleft, the synapse, and then the ligand class, being acetylcholine, attaches to its receptors class on the sarcolemma, that is the plasma membrane class of the muscle cell, and then that class is what opens up, I repeat, that class is what opens up those, of course, chemically gated sodium ion channels. I repeat, that is what then opens up, class, the chemically gated sodium ion channels. So there it is, class. The muscle cell will then depolarize and we'll get, class, what is called a nerve impulse, and then ultimately, class, contraction will ensue. So that will depolarize that cell, and it's all based on, class, acetylcholine, that is all based on, class, acetylcholine binding to its receptors, class. There. All right. This class shows much of the same that I just gave you here in another, of course, depiction. So G protein linked receptors, class, meaning those G protein receptors. Here, what we have is that outer part of the receptor has a binding site for a signal molecule, and the part of the receptor that extends, class, into the cytosol has a binding site for a specific G protein. So as it's like that, the receptor consists of 7 transmembrane helices that are connected by loops into the cytosol or the outside of the cell. So as this happens, just as such, you have the signal molecules, class, that bind, I didn't know that wasn't black, that bind here, class, and then from there, you, of course, get that G-linked protein, which you all see there. So the G protein binds to guanine nucleotides, and as that happens, class, when signaling molecules bind with the G protein, that G-linked receptor, the receptor, class, it changes shape, and as it changes shape, it allows the G protein to associate, class, with the receptor. There we see it there. So next up are enzyme-linked receptors, and we're just about done with this lecture. With enzyme-linked receptors, of course, they're transmembrane proteins with a binding site for a signaling molecule on the outside of the cell. So the proteins in this case, class, are called tyrosine kinases. To help you with that, class, a protein typically ends in ACE, typically. So of course, tyrosine kinases, of course, are a number, of course, enzymes. So as it happens, class, with that, the tyrosine kinase enzyme transfers the terminal phosphate group from ATP to a specific tyrosine part of a protein. A photo, class, will show it very well. So what happens, class, is that signal molecule binds to its receptors. There it is. So as that then happens, class, what we get is the conversion, class, from ATP to ADP. So those, of course, meaning that's using energy, class. I'll just say it right there. So our enzyme domain for the receptor is here. So we're now about to activate, class, the protein kinase sites. So what happens is it's dephosphorylating ATP, meaning using that energy to then, class, phosphorylate, class, the protein kinase sites, and they're now activated, class, by way of those phosphorylations, meaning by adding the phosphates. So I can't stress enough, before tyrosine kinase can phosphorylate a signal protein in the cell, it must first be activated. So that happens, of course, when those signal molecules bind to those 2 kinase receptors that I mentioned, class, there, and then the receptor proteins then move closer together in the plasma membrane to form that dimer, which is there. Thereafter, class, we get that change and take place. So each of those receptors, class, then add a phosphate from ATP. And thereafter, class, once activated, the tyrosine kinase enzymes can then, of course, phosphorylate the signaling proteins inside the cell. And there you have it. So now we've made it down, class, to signal transduction. I'll be here, class, for mere moments. So these are regulatory molecules that activate membrane proteins that then transduce the signal. So, of course, the signal molecule binds to that cell surface receptor class, it changes the shape, and then the signal class is relayed through that chain of protein kinases. So, of course, this is part of that signal pathway that amplifies the signal, as you all see on page 138, class with figure 6.6. Next up, class, and we're just about done, will be here with GABA. I'm stopping here at GABA class because it deals with the ion channel link receptor as it opens or closes. So we have a number of ion channel gates that remain closed until that ligand binds the receptor, such as the case class with gamma aminobutyric acid, known as GABA. So the receptors class bind to the neurotransmitter, GABA. And of course, what happens is that ligand gated chloride ion channels, meaning GABA then inhibits neural signaling when those chloride ions leave the cell, which inhibits transmission of neural impulses. I'm not going to give a lot here, class, but it's when barbiturates are taken, class, and even certain drugs such as Valium, class, bind to those GABA receptors. So that lowers the amount of GABA required to open the chloride ion channels, and that inhibit those impulses. So this results in that tranquilizing effect, meaning that's why people, of course, would take a tranquilizer that has been, class, I say prescribed, not taking class because people don't take those things. So with that, class, I'll say please make sure you prepare well for your test by knowing, class, what was presented earlier on in section 6.1 about the signal transduction pathway, class, and those 1, 2, 3, 4 parts being, of course, synthesis and release of that signal molecule, reception, class, of the information by target cells, signal transduction, and then ultimately, class, that physiological or metabolic response. This has been your instructor, Skylar Huff. Thank you all for listening and prepare well for your ensuing test.